Let's move on to our third segment, which is, a big and important segment, which is mCRPC, and really we have several really important issues to talk about because of this plethora of new medications that we're starting off early in mCSPC and in mCRPC. How is it influencing our decision-making, our discussions with patients when they get mCRPC? We've already touched to some degree significantly on the imaging sort of conundrum that will befall us now as we get more and more accurate, more sensitive scanning, how that changes our decision-making. But at this end of the day, too, we have so many life-prolonging agents really since 2010. So, Pedro, if you would, here's a sort of Herculean request, but if you could just review all the different therapies that have life-prolonging benefit in mCRPC.
I'll try to be succinct and kind of summarize how I do it with the patient, right? So once we have a patient who progresses to or develops castration-resistant prostate cancer, which we often define by either numbers, PSA progression, serologic progression, or by new disease in the scans, radiographic progression, or development of new symptoms. So once you develop progressive disease, doesn't matter how you define it, you have 6 life-prolonging therapies approved in that space. So you have 2 chemotherapy, both microtubule inhibitors, an older one, docetaxel, with 2 phase 3 trials, New England papers, ensure survival benefit there. You have cabazitaxel, which is kind of a cousin of docetaxel, a little bit different safety profile, and it's approved after the use of docetaxel. Which is tropic data to support their use. Then you have radium-223, which is a novel way of a bone targeting agent. Actually, as a matter of fact, is the first bone target agent to show a survival benefit in that space, which is quite remarkable and so for some of us a little bit unexpected, right? But we can dissecate that data a little bit better. We have a vaccine, Cipla-SLT, Prevent, which is actually, we're going to talk a little bit later today a little bit about immunotherapy, but just a reminder that we do have an immunotherapy agent approved for men with asymptomatic or mildly symptomatic castration-resistant prostate cancer, and then we have obviously, the AR target therapies with abiraterone and enzalutamide. Historically, it's good to tell the story how we developed all this, right? Because back in the days, more than a decade ago, we had docetaxel as the first chemotherapy agent that make people live longer. And so we classify all the drugs we developed as pre and post chemo. So we showed with Cook 301 and then affirmed that both enzalutamide and abiraterone would improve the clinical outcomes, including survival in that setting. And then we moved it early on in the course of disease, the pre-chemo space in the CRPC setting, with Cook 302 and Prevail, the same survival signal was present. So we have 6 life-prolonging therapies. There's a lot that's coming, and I know we're going to touch on that later. But if I had to summarize it, I would say we have different agents with different mechanism of action that allows us to control the disease for a decent amount of time.
Yeah, I mean, it's kind of, I guess, I'll just say some of us on the panel are old enough to remember where it was very nihilistic, where, patients with metastatic disease, it was ADT and really nothing else. And it's great to see that we've had all these, these advances that are really giving us challenges. And, the treatment of the patients has become that much more complex. And so there really has to be, I think, a real dedication to treating these patients and understanding what the data is and what data is also forthcoming. And why does it matter? Because, of course, we've touched on this issue around comorbidities and the nuances and the differences of the mode of administration and the tolerability of these drugs. But before we get more into that, William, you've done such great work on this. If we start using these drugs that typically get approved in the most end-stage patients, because it's been the pathway to approval of overall survival, you're either alive or you're not, and then we realize we'd have an opportunity to move these drugs up earlier in the disease continuum. But what's the impact on the disease biology?
Right. Well, as you know, when we started out, Neil, these patients didn't live much longer than a year or two at most when they developed castration resistance. and they are definitely living longer. But there's been some interesting and concerning statistics. For the first time in the American Cancer Society statistics, we're seeing a slight rise in the death rate from prostate cancer, and that's because we're not curing these patients. We're using these therapies earlier. The men are living longer for sure. They have a better quality of life. I believe all of those things. But the cancer continues to figure out ways to grow. And it's both very frustrating and very upsetting because despite all of the advances, these patients are progressing and unfortunately dying. And the reason probably has to do with biology. We're selecting for clones or subsets of cancer cells that figure out how to grow independent of androgen signaling, independent of the effects of chemotherapy, independent of the bone effects. We did a small study where we looked at the patterns over the last 20 years of where patients present with metastatic disease. When we started out, maybe on this side of the table, patients had bone mets 90% of the time. They still do have bone mets often, but they had lymph node mets 20% of the time. I remember some of the papers would say 20%. Now we know that it's like 50% or more. So we are shifting these patients to other sites of disease, maybe because we have better bone-targeted therapies, for example. So I think the biology will inform us. I think we have some new interesting targets where drugs are being developed that are important. We're going to talk a little bit about BRCA, but there are other targets of resistance, the neuroendocrine pathways. And the problem is that the drugs don't come fast enough. We may know that, for example, there's an important biological driver of some cancers, but we don't have drugs that target them well. But I do think that that's going to be the problem over the next 5 to 10 years, is as these patients get all of the therapies that Pedro just outlined, whether they're getting them very early or in the CRPC setting, we have to basically stop every single pathway that these cancer cells use to grow, and otherwise we're never going to cure them.
Yeah, we're starting to your point about, that's a really good summary, how do patients become mCRPC? Well, it seems like there's a disproportionate number that get into the mCRPC space. They come from either two buckets, right? They either start off with metastatic, as we talked about earlier, and then they progress, and now they're progressing and going to get a lot of other therapies before they get mCRPC. So what's the mutational effect on the adenocarcinoma cell, and how does it affect the aggressiveness of the biology and the resistance to lines of therapy? And then there's still the biochemical relapse patients who get ADT, get NMCRPC, and then move forward. And I think we're starting to see data because of our less screening over with the USPSTF. We're seeing where we historically said maybe 5% of patients in the US present with metastatic disease. We're starting to see data that suggests it may be north of 10%. Clearly in other parts of the world, it's as high as 50, 60%. So it's very interesting.